Good evening, guys. So today I'll be talking about the MCQ. I'll be reviewing the MCQs for the amino acids and protein lecture, also for methods of isolation. So question 1 says, which one of the following statements concerning the peptides shown below is correct? So this is the peptide here: glycine, cysteine, glutamate, serine, aspartate, arginine, and cysteine also. A says the peptide contains glutamine. No, it contains glutamine. So this is wrong. The peptide contains a side chain with a secondary amino group. It does not contain a side chain with a secondary amino group. The peptide contains the majority of amino acids with side chains that would be positively charged at pH 7. The only side chain that will be positively charged at pH 7 should be arginine. So doesn't contain many. So this is wrong. The peptide is able to form an internal disulfide bond. Yes, cysteine residues can form an internal disulfide bond. So that's the answer. Option D is the correct answer here. Question 2 says, which of the following acids play a major role in collagen synthesis? We reviewed that in the lectures today. The answer is glycine. Proteasomes are supramolecular assembly of enzymatic complexes that function in the degradation of damaged or unneeded cellular proteins. So proteasomes digest proteins. The peptide alanine, arginine, phenylalanine, glycine, glutamate is treated with peptidases. So the peptidases broke the bonds and separated them to release all the amino acids. The solution is adjusted to pH of 7. The electrophoresis is performed in the electrophoretograph. That's this electrophoretograph here depicted below. The amino acids indicated by this arrow should be what? So watch this. This is the negative electrode. This is the positive electrode. So positively charged amino acid will flow towards the negative electrode, and negatively charged amino acid will flow towards the positive electrode. The neutral ones will remain somewhere at the center here. So the one that could be here will be the positively charged electrode, and the one that will be positively charged at pH of 7 should be arginine. That's option B. Question 5 says, which of the following statements about protein structure is correct? The answer is, the information required for the correct folding of the protein is contained in the specific sequence of the amino acid along the polypeptide chain. Option E is the correct answer. Question 6 says, the addition of ammonium sulfate makes it possible to separate proteins from a mixture according to their degree of solubility. So that's what we call salting out. We looked at it in our lectures on X-ray crystallography. Which of the following amino acid is essential in the human diet? The essential amino acids are PVT, TIM, Hall. Hall is leucine and lysine, so lysine is present here. Hemoglobin electrophoresis is based on charge and mobility. That's the answer. Production of which of the following proteins will be most directly affected by scurvy? The answer is collagen. Which of the following types of bond is a covalent bond? A disulfide bond is a covalent bond. Identify both glucogenic and ketogenic. We said the mnemonics for that is 3TIP. So 3T stands for your tryptophan, your tyrosine, and your threonine. So they are the 3T. So we have one of them here. That's the option C. IP stands for isoleucine and phenylalanine for P. Question 12. The amino acids commonly used as an ingredient in the buffer of SDSPH. SDSPH stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. The amino acid commonly used is glycine. Glycine is able to have a negative charge at certain pH, a positive charge, and also a neutral charge. So it's the most ideal that we use for SDSPH to take out the charge from the electrophoretic film. Which of the following amino acids will be absent in an alpha helix structure? The answer is proline. Proline has a kink in its structure, so it cannot be found on the alpha helix. Question 14 says, using the structure below to answer question 14 and 15, the C terminal of the amino acid of the hexapeptide shown will be aspartate. Aspartate will be the C terminal and arginine will be the N-terminal. The answer is aspartate, option C. At physiologic pH of 7.4, this hexapeptide will contain a net charge of 4. So let's calculate it now. Aspartate is -1, alanine is 0, serine is 0, glutamate is -1, valine is 0, arginine is +1. So the summation of it will be -1. Option B is the answer. So this is it with the revision questions for the MCQs on amino acid chemistry, protein structures, and method of isolation and identification of protein. See you guys in the next lecture.